Walking in. So good morning, everybody. Thank you for coming. Welcome to the Great Lakes Seminar Series, which is co-sponsored by the Cooperative Institute for Great Lakes Research and the NOAA Great Lakes Environmental Research Laboratory. My name is Greg Dick, and I'm the director of the Cooperative Institute for Great Lakes Research. I want to welcome everyone in the room and everyone who's joining us online and everyone who might be listening to this at some point in the future. I hope the future is awesome. So it's my pleasure today to introduce Jenan Kharbush. Jenan is an assistant professor in the Department of Earth and Environmental Sciences at the University of Michigan. She's held that position since 2021. Before that, she was a presidential postdoc in our department, in the same department. And that's a really neat program that recruits top scientists as postdocs with the promise of a tenure track position following that. Before that, Jenan did a postdoc at Harvard University with Ian Pearson, and before that, she was a PhD student at the Scripps Institution of Oceanography, working with Lahini Alawahare, who's a chemical oceanographer and a geochemist. And Jenan is from a Great Lakes state, Wisconsin, and I can't remember what town, but I know Madison, so maybe just outside the basin is Madison. But I think Ripon College, where she did her undergrad, is in the basin, so a Great Lakes person here. And today she's going to talk to us about exploring the role of nitrogen substrate availability in the ecology of microcystis dominated CyanoHABs. So let's welcome Jenan.
Thanks, Greg. Appreciate that introduction. I actually never thought I would come back to the Midwest. When I left for California, I was like, I'm done with winter and I'm done with the Midwest. But yeah, it's super nice to be like back working in the Great Lakes, especially because when I was a kid, my parents actually tricked us for a while into thinking that Lake Michigan was the ocean. I really wanted to go to the ocean. So anyway, to introduce, I guess this doesn't work, I'll just use the keyboard. To introduce our lab, we call it the Microbial Biogeochemistry Lab. We often get confused with Greg's lab, which is the Geomicrobiology Lab. But we primarily are interested in kind of connecting scales. So connecting like what's happening with nutrient reservoirs, with the organisms that take up and transform those nutrients. And then there's a side of my research program that is focusing on what is happening with intracellular metabolic processes, including like synthesis of metabolites. And I'll talk a little bit about some of that work today. These are some of our current research directions. You might be like, wow, they're all over the place. And they are a little bit, but they are kind of united under this theme of either nitrogen, nutrient availability, and also CyanoHABs. So these are some of our lab members. I have several graduate students, and then some of you might know Ashley Bertner is the lab manager who runs everything. Okay, so I want to start with nitrogen, and this is probably this is a talk I gave yesterday, so there's extra background about nitrogen, but this is a major limiting nutrient for phytoplankton, for all life on Earth. Everything needs nitrogen, but in particular, it's something that phytoplankton need for growth, and so there is a feedback between the amount of bioavailable nitrogen on Earth and climate, actually. And when I say bioavailable N, I'm talking about nitrogen that organisms can use. So there's lots of nitrogen in the atmosphere as nitrogen gas, but organisms are not, very few organisms can take that and turn it into something that they can use to build their cells. So it's a limited resource because of that. The nitrogen cycle on Earth used to look like this. This is a figure from a textbook, and I've blocked some of it out. Oh, I have a laser pointer. So the main input of bioavailable or fixed nitrogen is going to be through biological nitrogen fixation. That used to limit the amount of nitrogen available for primary production to use. However, now the nitrogen cycle looks more like this. Humans have changed how much bioavailable nitrogen is in the biosphere through agriculture, and especially through industrial nitrogen fixation. So taking nitrogen out of the atmosphere using an industrial nitrogen fixation process. And what that has done is that it's essentially unbalanced the nitrogen cycle, where we are taking a lot of nitrogen out of the atmospheric reservoir and moving it into the biosphere. And a lot of that nitrogen is ending up in aquatic ecosystems. So it's causing, so these are some of the issues that have resulted, including dead zones and harmful algal blooms, which is going to be the focus of today. But as much of 50% of what we apply to grow crops is not actually used by the plants, and a lot of that then is what is driving harmful algal blooms. And because I don't want to get attacked, I always have to say that phosphorus is another major limiting nutrient, and it is the ultimate control on how much biomass, how much phytoplankton you get in lakes, including in harmful algal blooms. But worldwide, it is basically destabilizing both of these cycles that is the cause of harmful algal blooms. Okay. And so in Lake Erie, which here we are all very familiar with CyanoHABs in Lake Erie, these are often dominated by Microcystis, which produce the toxins microcystins. They're very nitrogen rich. So while P is important for overall bloom size, and that is how it is predicted every year, there's no phosphorus in the toxin. So the biosynthesis of this toxin is very strongly linked with nitrogen amount bioavailability. Okay. One thing that I'm interested in is it's not just concentration that matters for phytoplankton when they use nitrogen. The form of that bioavailable nitrogen also matters. To us, we often group it all together and say this is the total dissolved nitrogen that's available. But phytoplankton really distinguish between these forms. So this is artwork by my current PhD student. And we have kind of divided the phytoplankton. This is a generalization, but phytoplankton like cyanobacteria, like dinoflagellates, green algae, they tend to be better at obtaining ammonia and urea, so reduced nitrogen substrates, whereas things like diatoms are nitrate specialists. So these are often presented as like preferences that phytoplankton have. In reality, it's that they're adapted to like use these different forms and to outcompete other organisms that they live with for these specific forms of nitrogen. And this is ultimately driven by energetics. So the organisms that can take advantage of ammonia can have an advantage because ammonia is easily assimilated into biomass. It is the most reduced nitrogen substrate, and therefore it's most easily assimilated. Nitrate, on the other hand, needs to be reduced to ammonia before it can be built into cells. And so nitrate requires 4 times more energy for a cell to use it than ammonia. And, so why would organisms do this? They do it because there's less things that then are going to be using nitrate, and it gives them an advantage when there is, lots of nitrate around. And you see this like in the ocean or in Lake Erie, where the main form of dissolved nitrogen is going to be nitrate. And in fact, phytoplankton, this is a generalization. So phytoplankton can use one or more of these forms. And Microcystis, actually, because of course it can use all 3 of these substrates. But we don't know very much about like how much of each form of nitrogen it's using during the bloom, as nitrogen availability, so the amount and the ratios between these forms, that changes throughout the bloom. We don't know too much about how Microcystis adapt to the changing nitrogen bioavailability. So that's kind of the big question of what we've been working on in our lab is how does nitrogen availability, so like the total amount, but also the form, how does that influence Microcystis strain composition and its bloom ecology? And by bloom ecology, I'm talking about like, how it grows and toxin production, all of that, but also how it might interact with other community members during the bloom. And this is kind of how it's gone. it seems like you should be able to answer this question, especially given all of the data that we've collected over the years, like especially here. But in fact, this is what's happened. So there have been many pitfalls and excursions and side quests along the way. So I'm going to talk about a couple of those today. But okay, so first of all, why, nitrogen, we know it's used to synthesize the toxin, but actually not all Microcystis strains are going to produce microcystin. And this has been observed in several systems, not just Lake Erie, but there's often a toxic phase early on in the season, and then that progresses to a less toxic phase. And so this is my very unscientific plot from one of the blooms, just showing that you have cyanobacterial abundance, as indicated by phycocyanin, correlates with toxin abundance in July when the bloom is just getting going. And then later you see that toxin kind of decrease, but you still have very high cyanobacterial biomass that persists even past when toxin is not being produced as much. And that's still a problem. It's still gross, and they make other things when they're not making the toxin. So we still want to understand, what's going on in that later part of the bloom. At the same time as this is happening, we see a change in the nitrogen availability. So you have a decrease overall in the amount of nitrogen that is there, and it often ends up being nitrogen limited at the end of the summer. And if you look at the form of nitrogen, what's there is mostly organic forms of nitrogen by the time you have the decline of that toxin production, but you still have a lot of cyanobacterial biomass. And we know, thanks to some work in Greg's group by his former PhD student, Colleen Yancey, that this is not just that they're like turning off the genes for the toxin production. They are actually, there's actually a strain shift. So there's a shift in the toxic to non-toxic strain dominance. I'm sure a lot of you have seen this paper. But there's actually 3 kind of genotypes for the microcystin synthesis gene package. Sorry, I'm not at my best, so sometimes I'm going to forget the words here. But we use the MCY gene, and so that's what this is showing. And so there is a absent where they do not have the genes to make microcystin. There is a partial where they make like half the microcystin molecule, and then there's a complete pathway. And so this figure is just showing that at different stations, you see that you start with a complete pathway, and then there's a shift to either a mix of like partial or the absent genotypes. This is also important to understand because, again, recent work by a former grad student of Greg, Lauren Hart, we looked at this like shift from inorganic nitrogen to when you have more organic and processed nitrogen present, and she correlated gene abundance, but also with the metabolites that you see. And there's a transition from high nitrogen requiring things, which includes microcystis, but there's also other things like anabenopeptin, that are synthesized through what are called non-ribosomal peptide synthase pathways. And these are essentially like giant enzyme complexes. So to build these, not only to make the toxin, but to build the enzyme complexes, you need a lot of nitrogen. And so you see those showing up repeatedly, like in the early parts of the bloom. And then there's a transition to things that are synthesized via ribosomes. So these RIPs and PKS molecules, not only do they contain less nitrogen, but the machinery needed to build them contains less nitrogen. So over multiple years, you can see these patterns, which is very cool and indicates that we don't just have to worry about nitrogen from the perspective of microcystin production, but there are all of these other metabolites and toxins. Well, we don't know if they're toxins, some of them. So there might be these other things that are being produced that are also affected by nitrogen availability. So that's just some additional justification for why you should care about this question. And we've been kind of coming at this from a bunch of different angles, but I'm just going to talk about 2 of them today. The first project is something that we have now published, but we we're thinking about like, okay, as the bloom is progressing, what is happening with microcystis and the things that live with it. So we're going to look at exometabolomics. And then the second project is going to look at in the field, what happens with if you give microcystis different nitrogen substrates at different bloom stages, how does the amount that they uptake change? We're not going to answer the big question today, I'm sorry, but the smaller questions. Okay, so for project one, just some background. We did a study in collaboration with some scientists at Lawrence Livermore, who I will introduce in a little bit. But we looked at how the microcystis associated like microbial communities might affect nitrogen uptake. So there was a study by Derek Smith in 2021 where he found that the microcystis microbiome actually changes between strains and over time during blooms. So there is a microbiome that is associated with certain strains, and when the strains change, so does the microbiome. And then the study that we did in collaboration with Lawrence Livermore, this Lee et al. Study, we added labeled substrates to cultures and looked at how the heterotrophic cells associated with them helped process that nitrogen, including organic nitrogen. So we gave them labeled amino acids, and then we could see that they were taken up by the heterotrophic bacteria, and some of that nitrogen eventually made its way to microcystis. So some of these microbes that are living together with microcystis are probably helpful, especially like later in the bloom stages when nitrogen gets like really limiting. But in early bloom stages, we're not sure what they're doing. Are they helpful or are they maybe competing? So maybe that role of bacteria shifts during the bloom. And so this is just a microscopy image, like in case a lot of you maybe have seen a colony of microcystis and looked at what's in it. But the orange round cells are microcystis cells. The little tiny, there's like some tiny ones in here. I don't know, maybe these are cyanobium. I don't know that we ever determined it, but there's some other kind of small cyanobacteria. And then all of the green dots and things in the background, so the very tiny things, those are all heterotrophic bacteria. So these orange cells are actually embedded in like a sticky exopolysaccharide matrix that these other organisms are also living in. Okay, so this is answering this question of how might microcystis interact with these things, its friends, its frenemies that live in the phycosphere around it. The phycosphere is this like region around the cell. It was coined in this Seymour 2017 paper. The region where you have an enriched concentration of organic stuff like exuded or produced by the host cell that bacteria can probably use. And so again, like we know that this phycosphere community changes between microcystis strains and over time. In this paper, they also found that the metabolic capabilities of the microcystis strains seem to affect what heterotrophs were there. And so that kind of led us to, we wanted to know more about like, well, what does microcystis produce that bacteria might be able to use? And does that change depending on what strain of microcystis you're looking at? And when I first started thinking about this, I was kind of surprised that we know almost nothing. There's like 2 papers that look at the exometabolome of microcystis. But based on the previous work with the Lee et al. Paper, we knew that heterotrophs, like some of them at least, were processing organic nitrogen. Okay, so this is work by an undergrad, actually, shockingly. One of the best that I've had. And so she did this project where she chose 3 microcystis strains to ask the question of whether different nitrogen substrates would result in changes of what microcystis produced. And she chose the strains based on different characteristics. She wanted to compare as many variables as possible. And so we picked an azenic kind of a type strain, like a lab rat strain. That's the PCC 7806. It makes toxin. So it has the full MCY gene package. And then we chose one of the cultures from the Western Lake Erie culture collection that had the partial MCY genotype, and we picked one that had no MCY. And both of the ones from the culture collection have heterotrophs associated with them, so they're not azenic. And then we also looked at whether those heterotrophs had the known microcystis degradation pathway, in case that made a difference in what microcystis might produce and what we might find in the media. You don't have to remember this really because none of it matters, turns out. So we had this hypothesis that yes, we would see differences because the energetics of the nitrogen substrates that they're assimilating would affect like what metabolites they made, in addition to things like whether they were producing toxin or not. And there was a paper, I think by Lauren Krausfeld, that we kind of built off of because they also looked at what happened with the metabolic pathways internally, and they saw differences in where their nitrogen label went. But they were looking intracellularly, and this is looking at extracellular stuff. So the methods, I won't spend too much time on this. It's what's called an untargeted metabolomics workflow. Lauren Hart helped us a lot with this, but we used liquid chromatography, tandem mass spectrometry data. So essentially you're separating your molecules with liquid chromatography, and then the mass spectrometer is breaking them apart into pieces, and you look at those pieces to figure out what molecule it is, or more likely what type of molecule it is, because it's very hard to identify things from the environment via untargeted metabolomics. So there's a library through this GNPS database that you use to detect like known compounds, and then but a lot of it is just annotated as a type or a class of compound using a tool called Sirius. So it essentially looks at the fragments and says, this fragment looks like an amino acid fragment, so therefore I'm going to put it here. And it gives you a probability of, how likely is that prediction to be correct. And so we detected 275 total across all the treatments. And we only had 11 library hits, which is like a womp womp moment. But that's pretty typical for the number of compounds, like the relative amount of compounds that you can identify in environmental samples. And then we did a bunch of statistics to figure out what could we learn from this. So these are the library hits that we see. The toxin is there. That's good, right? Picked up the toxin. We saw aerocyclamides, annabinopeptins, and then D and E are sunscreen compounds. And I won't talk too much about those today, but they had some interesting patterns that you can go and look up in the actual paper. But we focused mostly on nitrogen-containing things because that is, maybe not unexpectedly, that is what was really driving the differences between our treatments. So we did kind of a principal component analysis where we looked at like what is driving the differences between treatments in terms of the types of molecules that were identified. And so here we can show there's like two things that really separate it. So this is the media in the gray. So these are all the nitrate treatments. These are all the reduced nitrogen treatments. So ammonia and urea kind of clustered together. And that makes sense because they're both reduced sources of nitrogen. The inputs are into the cellular assimilation pathway are more similar than for nitrate. And then you can see that the strains also cluster together. But the clustering is more distinct when they're grown on nitrate, whereas there is some overlap between the partial and absent MCY genotypes when they're grown on reduced nitrogen. And then we kind of separated out what were the most important factors. And so strain is still the most important factor that is driving the differences between the types of exometabolites that were in each treatment. But then the second important factor is substrate. So this is supporting the hypothesis that yes, when you give them different sources of nitrogen, they are going to produce different sets of compounds and they're going to release different types of compounds. This is an awful figure and I really don't like it. And you can't really see much on the screen probably. But this is just another way of looking at what I just showed you, but this figure has compound class information. And so like the Sirius assigns these compound classes. And so like we kind of tried to highlight like, okay, here are purines and pyrimidines across all the treatments. But what I want you to notice are that, so these are the same strain grown on different substrate treatments, and they are clustering, just like we saw in the previous figure, by either reduced nitrogen or oxidized nitrogen in terms of what metabolites you see in the media. And the same is true for the other strains that we looked at. The most abundant compound classes that we saw were by far amino acids, purines, pyrimidines, and then other nitrogen-containing compounds. We're all, and part of that is like expected, like we're giving them a lot of nitrogen in these experiments, even though they're different types of nitrogen. And so they probably have excess, and that maybe affects what you see in the media. But another important factor that I don't often see people admit in talks or manuscripts is that your mass spectrometry method can bias your results. And so we used only positive ionization, so that adds a positive charge to the molecule. In mass spec, you only see a molecule if it's charged. And so we didn't do negative mode mass spectrometry. If we had, we might have seen different distribution of compounds. So we are biasing our results a little bit by using positive mode. However, that's what's going to ionize most nitrogen-containing compounds, which is sort of what we're interested in. So small like caveat there. The nitrate treatments were more distinct than the ammonia or urea grown cultures from the same strain. So that's what I was trying to show by circling the boxes at the top, is that the nitrate, even though they're the same strain, they produce very different compounds when they're grown on nitrate. And also when you grew the strains on nitrate, they produced the most diverse exometabolomes. So they produced like the highest number of compounds that were across these like subclasses over here. And then finally, there were no compounds that we could find that were produced only in ammonia or urea. Like whatever compounds were detected in ammonia were also detected in the urea treatment. So again, this is reinforcing that there is some kind of energetic driving force behind these patterns. And I'll say a little bit about the toxins because that's what you care about. There were some interesting patterns in our study where we saw microcystin and aerocyclamide. Those were only detected in the lab rat strain, but they were detected at the highest abundance on nitrate. And it's good that we only saw microcystin in the lab rat strain because that is the only one that had the full toxin set of genes. And then kind of on the other end of things, we also detected anabenopeptin, but only in the non-toxic strain. So non-toxic meaning non-microcystin strain, but also at highest abundance under nitrate amendment. So this is suggesting that maybe the same nitrogen conditions that would promote production of microcystin might also promote production of these other things that are synthesized by similar like enzymatic pathways. Unfortunately, like this agrees with some of the prior work on the Microcystis exometabolome, and it disagrees with another paper. So there might be some strain variability that we're not capturing here with our 3 selected strains, because those other studies did use different strains. Okay, so I cannot see the clock. I have no idea. Okay, yes, I'm doing okay. So the ecological implications of this particular study, even though this is sort of just, I feel like opening Pandora's box maybe, are that like the changes in the type of N as the bloom goes along could actually be affecting what they make and what gets exuded for their friends to use. And we did kind of show that the urea and ammonia, the reduced nitrogen treatments, are producing an exometabolome that is more similar than to the treatments grown on nitrate. And what was driving those differences were differences in the amount of extracellular amino acids and nitrogen-containing things like purines. But there were also fatty acids that we saw showing up more in the nitrate treatments than in the others. And implications for like, why does this matter? Well, these are really yummy molecules for bacteria. So it is potentially like an extra nitrogen source for their heterotrophic affiliates or symbionts. Another thing that I thought about was that the most abundant nitrogen substrate during bloom season is nitrate, especially when you're making the toxin. And so during that time, maybe bacteria, a lot of which cannot use nitrate, bacteria that can associate with Microcystis during the earlier bloom stages might actually gain an advantage because they could get at these like nitrogen-containing exudates that Microcystis produces more of when they're grown on nitrate. And future work, things that we didn't really consider, but which I think are probably pretty important. If I can like suck in another student to work on this. Light, I think, probably really matters. We just kept the light at, the whatever the standard, not too low and not too high, so that they would be happy. But I think that there might be differences in the energetics of what substrate they use and how they use it, depending on light. Because in other cyanobacteria, the use of ammonia versus nitrate depends a lot on energy and light availability. And so maybe the same is true of Microcystis. They can use all three. And so like if you think about the scums or, they float on the top in Lake Erie, they get a ton of light. Maybe they're not energy limited at all. And so in that situation, maybe they actually prefer to use nitrate because they have plenty of energy. And that way they don't have to compete for the less abundant substrates with all of the other things that live in the community. Okay. All right, so that's like kind of project one, getting at this big question. So I'll switch a little bit gears here and talk about the second project. So that was a culture study. This is a study that we wanted to do in the field. We did this as kind of a follow-up to the Lee et al. Study, which used cultures. We wanted to look at how the cell-specific uptake of nitrogen worked in natural communities. And so this has been a while. We did these experiments in 2021, and we wanted to get to the different stages of the bloom. So again, here's a crappy graph showing how we tried to target what we called early bloom. So before the peak in microcystin, which is the pink line here, we wanted to get a midpoint, like sort of when toxin is maximum, and then a late time point when toxin production has declined. And we did pretty well. It was hard because it's hard to predict how the bloom is going to go every year. And then we took that whole water and we added nitrogen N15 labeled substrates. So we looked at ammonia, nitrate, and urea. I think we had an amino acid treatment, but it didn't turn out very well over the 4-hour time period that we were looking at. And we kept the time of incubation short because we only want to look at uptake here. And we don't want to have any recycling or exchange as much as possible between Microcystis and the other things in the community. So we used nano-SIMS as a tool, and I'll explain a little bit more about nano-SIMS in a bit, to allow us to measure uptake rates of these different substrates by Microcystis versus other things in these different bloom stages. And then we just got some omics data back. So we have meta-G and meta-T that is being used to complement the nano-SIMS data. And so these are our collaborators at Lawrence Livermore, Xavier Mayali, who runs the nano-SIMS, and then a postdoc, Dr. Wei Li, also helped us a lot with this. And then I also want to acknowledge all the people, because this has gone on for so long, there have been many undergrads that worked on this and processed all of this data that will be on the final product, as well as, so Dr. Sarah Rivera was Greg's lab manager until recently, and then Dr. Anders Kieladal is now the co-director kind of Greg's lab. And so he's been very involved in this. And then Lauren Hart, who worked on a lot of stuff. She was involved in a lot of things. So this is a lot of people. It's a lot of data. So this is what the 2021 bloom actually looks like when you look at all the stations and then look at the average. So on the top is the particulate microcystin concentration. And thanks to many of you, maybe, were any of you here for 2021 and collecting data? I don't know, but we owe all of this to Noah Glurl, actually. So, but this was like a moderate bloom. It was like a 6 on the, oh, I guess it goes up to 11, the scale. But so it had a very large spatial extent that year, but the biomass itself was not very dense. And so unfortunately, we didn't sample the same station because we were kind of getting samples opportunistically. It was the year after the pandemic. So this is not, it's a good example of when your study is good, but it is not perfect. And so the early time point comes from a different station. It comes from a station where there was a visible bloom of Microcystis. The mid and late time points come from WE09. There was also a visible bloom, but I think it was less dense than the WE16 near station. So the colored dots, I'm not sure if you can see them, but are water samples. So you can see that here, this one, compared to like the average, the early time point here had a very high microcystin concentration relative to the average. But the other variables, nitrate and nitrite, kind of were similar to what you see across the stations. And again, like this sample just seemed to have a lot more, like a denser population of Microcystis than you saw on average at the other stations. So looking at the community composition between the time points across all of these, Microcystis is the major cyanobacterium present, but then as you get into the later bloom, some other stuff starts to show up. So cyanobium is one of the ones that you often see with Microcystis, and then Dolichospermum likes to show up like in the fall-ish when things start to cool off and when you have a lot less nitrogen present. And so this is via metagenomics. And then also via metagenomics, we looked at the strain composition of Microcystis, and there's not like a ton I can say about this yet, because I haven't familiarized myself with all of the clades. The main thing is that they do change over time, but Microcystis genomics is really messy. So the clades are not like clean, where a clade has a set of defined characteristics. They're often really mixed up. But the purpley blue ones up here, these are ones that are known to do well at low phosphorus conditions. The MA3 clade, which are the gray ones, those were some of the strains present during the Toledo water crisis. So there's a lot of toxin producing strains in those clades, and they decline over the bloom, which is good because if we look at the MCY genes, we see that there's an increase and then a decrease. So we are capturing what we wanted, where we've got a before, a during, and an after toxin production. And there's some clades that increase, like the Microcystis, I can't remember, Floriaceae or something. Those contain both toxigenic and non-toxigenic strains, and so like we also see those in the middle and late bloom samples. So there is a shift in strain composition over time, and it seems to correlate with what we see in the toxin gene data. So this is new data. I have a master's student who's converting into a PhD student who's been learning omics. And so this is like very preliminary analysis of the data that we've recently received. So I would love any feedback actually on this data because it's very new. But essentially, you can think of this data as like who is there and what they were primed to do, because we don't have a time final. We basically have a meta G and meta T of the water before we did anything to it. So the community that was there. And so at first we looked at meta G and we just focused on nitrogen assimilation and then transporter genes. And so the way that this is oriented is you're looking at ammonium, nitrate, and urea, and then the sorry, this is hard to manipulate. We're progressing through the bloom this way. And we're looking at Microcystis, we're looking at other cyanobacteria, and then non-cyanobacteria. And so it's not super interesting. You can see that ammonium is really important for everything that's not a cyanobacterium, which is expected. So the majority of the genes in the metagenome that are ammonia assimilation and also transporters belong to non-cyanobacteria. And then Microcystis makes up a good portion. I think this decrease, even though this is normalized to the total number of genes, I think that this maybe reflects abundance of Microcystis versus, because we haven't normalized this to any particular gene. Okay, and I do have to say also that this makes sense because most Microcystis strains have all the nitrogen assimilation genes. Okay. So more interesting is when we get to look at expression through the metatranscriptome. So this is just to remind you what the metagenome looked like. And then this is transporter gene expression, and Colton divided this into the different genes in case you are someone who knows what the different genes are. But if you don't, again, we're looking at ammonia, nitrate, and urea associated transporter genes. And so what you can see here is that expression of these genes is dominated by Microcystis. So they are the ones that are expressing these transporter genes and that are probably like actively taking up a lot of the nitrogen that is being added. This is not a flat line, so there is a change in the late bloom time point where this, like let's take this as an example, urea transporters, this increases from the mid to late time point. But there's very few transcripts, so our error bars are kind of large. But in general, it's Microcystis that is expressing all of these genes. And then here are the assimilation genes. So again, a similar pattern. You've got Microcystis expressing a lot of the nitrogen assimilation genes. We added central nitrogen metabolism here. That is the GS-GOGAT cycle for which the input is ammonia and the output is like amino acids, glutamine and glutamate. And that's what all phytoplankton in this study are going to be using to assimilate nitrogen. And I don't know what to make of that yet. It's a bit strange that one of the genes involved in that is very highly expressed and the other is not so much. But what I think is really interesting here is that urease is not expressed very much at all. Again, this is not zero. It is there. And so maybe like Paul and I should have a conversation about whether we're doing this correctly. But I think actually this is correct. And the nano-SIMS data is going to show why. Okay, so to summarize these omics results before I jump into the nano-SIMS, the non-cyanobacteria clearly rely on ammonia. They have a lot of those ammonium acquisition genes, but then when you look at who's actually expressing the genes to take up ammonia, it's still mostly Microcystis, and they dominate expression of all of the nitrogen-related genes. And they also express all the nitrogen substrate transporters like all the time. So that was kind of surprising to me. I expected to see a little bit more variation, but they seem to just be ready to like take up, because there's no nitrate in the late bloom typically that's present, and yet they're expressing all of the nitrate acquisition and assimilation machinery anyway, which to me that's surprising because that is energetically expensive. But again, maybe they're not energy limited if they're just sitting at the surface with all of that light. Yes, that is what I just said. And then this urease thing is kind of surprising because the expression of the transporters are there, but not urease, even though they have them in their genome. They have urease in their genome. So either this is maybe regulated differently, where it's not regulated at the transcriptional level, or, and I think it's the latter, urea is being hydrolyzed externally and not by an internal urease enzyme. Okay, and then there is evidence of nitrogen limitation in the bloom stage. It was hard to see, but you can see an increase in all of these genes in the late bloom across Microcystis, the other cyanos, and the heterotrophic bacteria. And there were a few nitrogen fixation genes that we detected as showing up in the late bloom. Okay, yay, I left the exciting part till last. Okay, so like I think a lot of people have never like heard of or used nano-SIMS, so I'm going to explain a little bit about this. Nano-SIMS is a really powerful technique. It can measure isotopic composition down to like 1 micromolar resolution, which is about the size of a cell. So you can use this to examine nutrient uptake and exchange as long as you use like an isotopic label. It's really good at tracing where that label goes. So this was first used like in the deep sea to look at exchange of nutrients between like, first using the environment in the deep sea to look at the exchange between like archaea and associated like sulfur bacteria. And so those studies were doing really, really fine scale. You know, there was a one cell that was the archaea and one cell that was the bacterium, and then looking at nutrient exchange. Our study is very different. It's looking at uptake by many, 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 many cells in the environment, and then trying to kind of converge on a average and look at like large long-term trends. So this is why this is a lot of work. So when you do nano-SIMS, you get an image like this first, which looks a lot like an electron microscope image. So these like cells are all microcystis cells. And then it can look for certain isotopic ratios. So it measures, this one is 13C abundance and then N15 abundance. So everywhere you see like a bright red or warm color, that's where that isotope label has been incorporated. And so this is from the Lee et al. Study. It's where we added dual labeled amino acids. So they're labeled with 13C and 15N. And so if you see that being incorporated into the cell, it means they've taken up the entire amino acid. So in the 13C image, you can see these bright red spots. These are all associated heterotrophic bacteria. You can see that there's some similar patterns over here where they've taken up the N15 as well. So like this cell, for example, here has taken up both, meaning it has taken up the entire amino acid into the cell. If you were to see a cell where they only show up as N15, so I don't know if I can find one, this one, oh, it's the opposite. It only has C13. I don't know if I can find one. But what that would indicate is that something broke apart the amino acid outside the cell, and then it only assimilated the nitrogen part of that molecule. And so this is going to be important when we get to the urea discussion. But this is the data that you get, and the software will automatically select regions of interest, which end up being bacteria. It does not select the microcystis cells, and so the role of all of these undergrads was to go and manually circle these microcystis cells for analysis. So it's a lot of work. And it's been pointed out to me that there is likely a way to automate this now, and so maybe that's what we'll have to do next. But it was a very good undergrad project. So here's the results for just microcystis cells. And we added several concentrations of the different nitrogen substrates. So this is the highest concentration that we added. So these are like the highest potential uptake rates by microcystis. And so what you're looking at here is the early, middle, and late time points, and then the 3 substrates that we added are down here. And then this is called NNet, and it's essentially taking into account that there was ambient nitrogen in the water when you added the labeled substrate, and the cells don't really distinguish, they take up all of it. And so if you don't account for this, you're going to be underestimating how much nitrogen they took up during your incubation period. So nnet is essentially telling you after the 4 hours, the percentage of that cell that consists of labeled nitrogen. So it's just a way of normalizing. So these are therefore like comparable. We can compare the early to the middle to the late in terms of amount of uptake without worrying about the fact that in the early bloom there was more, like nitrate, for example, than in the late bloom. So it's a way of normalizing. Okay. So there are like hundreds, there's probably thousands of data points in this data set, which is part of the reason why it's taken so long. But there are some trends that emerge here. And one of those is that the microcystis cells incorporate a lot more ammonium than urea or nitrate at all the bloom stages. So that is their favorite thing. And that is consistent with previous work showing that there are really high rates of ammonia uptake and regeneration during blooms. And so a lot of what microcystis like is ammonia, but they are also taking up the other things. So it's backed up by the omics data. They will use anything. We also see that there's an increase in the amount of ammonia that gets incorporated in the middle to late bloom stages compared to the early bloom. And this could reflect that it's more N-limited toward the middle and late bloom. And so maybe they express more transporters. This is something I'd like to investigate in the transcriptome data, is dig a little bit deeper into the AMT family, which is what I showed plotted. Has a bunch of different ammonia transporters within it, and some of them are high versus low affinity. So maybe that is something that changes in addition to the strains that are there. And probably ammonia is their favorite because it's the least energy intensive, and they're really good at obtaining it if they're expressing all of these uptake transporters throughout the bloom. Okay, and then we looked at these other concentrations. So we also added 2 micromolar and 0.5 micromolar. And the idea behind this was that we could tell if they were saturated for that particular nitrogen substrate. So if they were at their limit for how much they could possibly take up and assimilate. So as we go from 0.5 this way to 20, did they take up more? If they did, that means that their transporters, their machinery was not saturated. But if they didn't, then they were saturated. And so there are changes. So now I've switched this up on you. Over here, we have the early, middle, and late bloom. And then the different substrates now are across the top. But there is a shift in particular in ammonia where they are saturated in the early bloom, maybe because there's plenty of nitrogen around. So they don't take up more when you add more. But then later on, you do see that when you add more, their uptake increases. Weird stuff also happens, like if you look at the background kind of dots, these dots are all individual cells. So what I'm showing are medians, but there's quite a bit of variation in these microcystis cells in terms of like how much they take up. And I don't know, like one thing I would like to test is like, these are colonies. Is there a difference in where they are in the colony? Like that would be super cool to know. But you don't see it all the time. Like this, in the early bloom, they are all kind of concentrated around the median. And then there are changes in it. So there's changes in like the number of cells and how much they take up. The other substrates like urea and nitrate are taken up at about the same order of magnitude. And I found this surprising because in the, exometabolite study, we definitely saw that reduced nitrogen was kind of treated similarly once it was inside the cell. But here it seems like they're not taking up more urea just because it's a reduced form of nitrogen. And maybe that's going to make sense in a bit. And then in terms of saturation, they were only really not saturated like in the late bloom. And again, I think that has to do with how nitrogen limited they are. Okay, I don't have that much time, so I might just go quickly over the non-microcystis cells. The main difference is that they're similar in that they like ammonia more than the other stuff. This includes heterotrophic bacteria, but it also includes cyanobacteria that were not microcystis because we stupidly did not add 13C bicarb, which would have allowed us to distinguish. And in our experiments that are ongoing now, we are definitely doing that to be able to distinguish all the autotrophs from the heterotrophs. So some of this, like the nitrate and urea uptake, might be other cyanos that are just really small. But the main thing is that there's a big range in the amount of nitrogen that they can take up, and that just has to do with there's a lot of different organisms in this fraction of the community. And I'm just going to skip that because it's not very interesting and go on to this. So then we took this data and kind of wanted to know well, when we add the nitrogen, where does it go in the community? And does that change depending on the concentration that you add and what stage of the bloom that it is? So now we've got concentration across the top and substrate here. And then you're going from early to late bloom. And so Microcystis is really dominating all of the nitrogen uptake in that early bloom sample. And I just think part of that is based on what and where we sampled. I think that community was mostly Microcystis, so of course they're taking up the bulk of the nitrogen. But then in these middle and late bloom stages, the rest of the community takes up more of it. And in particular, the ammonium, there is a change in how much the rest of the community takes up depending on the concentration that you add. So Microcystis doesn't seem to do as well when you add a low concentration of ammonia versus when you add a very high concentration, they're like, great, and they just suck it all up. And nitrate is a similar pattern. And so essentially the rest of the community becomes more important in nitrogen uptake in those middle and late bloom stages. So that's kind of expected, especially if you have a lot of recycling happening when everything is nitrogen limited. And I forgot to say this, but the way that we did this is not very good. So I'm hoping to make this a little bit better. But I just took the abundance data for Microcystis versus everything else from the metagenomes. And then I just made probably a bad assumption that Microcystis have 10 times more nitrogen than all the other cells based on their cell size. And then you can use the average N net for each sample to figure out, estimate where the nitrogen is going. Okay, so conclusions from this, they're maybe not very conclusive yet. But ammonia is definitely taken up more by Microcystis, so they seem to prefer ammonia. I reviewed a manuscript recently where they would not agree with this. They found that, and I'm only saying it because I think it's going to come out soon. It's been accepted, but urea was the favorite substrate in their experiments. So that's very interesting. Even though ammonia concentrations are very low throughout the bloom, you only see the saturation of their transporters very early on. And so that's interesting in that it maybe suggests that when we look at the different AMT genes, the different ammonia transporters, maybe we will see some differences in affinity of the transporters that are being expressed. So yeah, Microcystis are really voracious consumers of ammonia. And so that's also maybe why when everything is nitrogen limited and everything has to rely on recycling, they're still winning. They're still blooming, even when you run out of most of the other nitrogen. But they also really like nitrate. And yes, so we saw kind of nitrogen limitation, and maybe this is just more support for the idea that in the late bloom, you're moving toward a more heterotrophic, a stronger, to use an oceanographic term, a stronger microbial loop, microbial recycling of nitrogen in the late bloom stages. So yes, we're going to explore that. And just a last note here about urea, because I'm kind of obsessed with urea. So there was this previous study that showed that Microcystis could incorporate both nitrogen and carbon from urea, and maybe that would make urea even worse. We shouldn't have urea in Lake Erie because they can get 2 nitrogens and a carbon from 1 molecule of urea. The nano-SIMS data, we use dual labeled urea, and we don't see incorporation of that 13C. So this is kind of in agreement with our omics data, which showed that they have the urea transporters, But they don't express the urease genes. And so maybe that's because they're not actually taking up the urea molecules. They are getting the nitrogen from the environment after the urea molecule has been broken apart by bacteria, although it's such a short time scale that that's hard to believe. Or there are external hydrolases that are in the colony matrix or in the water that are breaking apart the urea, and then the microcystis are using just the nitrogen. So that's what I think is happening. But we would like to look at the metatranscriptomes for additional evidence of that, and I'm not sure what we even look for. So maybe someone has a suggestion. But we also have been doing some ongoing, some more nano-SIMS experiments where we're trying to reproduce what they found in the Krausfeld paper. They incubated for 7 days, which to me is a really long time. And so even if they could have had external urea hydrolysis happening and carbon being released, and then over 7 days, that labeled carbon would be taken up by the cells as bicarbonate. And so you would see incorporation of both. So we've been trying again and doing some shorter and some longer term incubations to see if that makes a difference. Yeah, so okay, with that, I think like I made it in an hour. I have to thank Noah Glorl and Greg's Western Lake Erie Culture Collection and our collaborators at Lawrence Livermore's, and then all of the undergrad co-authors and funding sources. So, and then hopefully I can take like a couple of questions, but I don't know. Yeah.
Yeah, we will take a couple questions. It's, I think, right around 1. So if folks need to take off, of course, feel free. But for those that can stick around, we'll accept questions. And I see Reagan has the first. Margaret's walking around with the microphone, so let's use that so the folks online can hear us. And if you're online, please feel free to type your question in the chat. Thanks.
Thanks, Jenan. This was wonderful. Great work. Going back to your first, there's lots of questions I have, but I'll save them for later. But there's always been this question of where microcystis is actually located, whether or not it's in the cell or in the extracellular matrix. And we just call it all particulate versus this dissolved. How much of that LR that you saw was actually, did you also take totals so that you could see how much was in the extracellular?
No, and I wish that we had, but yeah, so these are some of the limitations of having this be an undergrad project, is that we were kind of time limited on what we could do. But yes, that would have been an excellent thing to look at. So yeah, I didn't say, but what we're looking at in the exometabolite and the exometabolome is not just things that they export, it's things that are like released when they die. And so like there are things like the toxin that maybe are just getting released when they die. There's another undergrad thesis that is not published, but we also took the cellular fraction, and then she analyzed the intracellular metabolites, and the patterns are different. So it's not just that we're not just looking at like a shadow of what's in the cell. It's stuff that gets released, but then yeah, also stuff that dies. And the way that we like justified it to reviewers was that regardless of how it happens, like cells are dying in the environment too. Yeah. So either way, like whatever's being released through cell death, that also matters because things will use it. Yeah.
I'm curious of like if they're actively giving it to their buddies in order to then disperse something else to give back to them.
Yeah,
you know what I mean?
I don't know. And I think that's something that Anders is working on with the microcystin degradation pathway, because sometimes they see degradation even when the pathway isn't there. So there might be other degradation pathways that we don't know about yet. So it could be like some kind of currency that they exchange. Because there's a transporter too, right? A microcystin transporter that nobody really knows what it's for or how it works. So
cool. I'll save my other questions.
We do have one online. So this is from Sylvia. Nice talk. Do you think the heterotrophic bacteria in the biosphere, sorry,
Icosphere, yes, good.
might be making extracellular urease hydrolysis enzymes?
Yes, I think that's a possibility. But that is something we should see maybe in the transcriptomes. But this is, so omics is not my area of expertise. And so I kind of don't have a good handle on whether we should expect to see that or whether it's going to be of too low abundance to detect. But that's something that like you would think that you would look for, because that would make sense. That maybe is benefiting the phycosphere community for them to produce those external ureases.
There's another one. It's very long. Sorry. Trying to read ahead. Okay. You mentioned that the nano-SIMS shows spatial data and your data has early, mid, and late bloom. But I was also wondering about spatial dynamics within a bloom. With a thick scum on the surface, the light attenuation with depth is also very stark. So I wonder if there may be some vertical stratification of end-use strategies based on light availability, even within a few centimeters down. Maybe hard to capture, but it would be cool somehow. Thank you.
Yes, I agree. I agree with that. And maybe my next obsession is going to be with light. We're sort of looking at this, but with another project in the winter where we're looking at light attenuation and how that affects nitrogen usage by the phytoplankton community. And so maybe we could try to incorporate this also in our summer sampling where to get at that question. But yes, thank you. That is a very good idea. And let people go.
Yeah.
Yeah.
Well, I have lots of questions, but I'll save them for lunch, I think. So I just want to thank everyone again for coming, and let's thank Jenan one more time.